Assalamu alaikum, everyone. My name is Samra Mirza, and I'm a third-year medical student. For today's session, we're going to be doing a histology crash course. This should be sufficient for you for your lectures of epithelium, connective tissue, and lymphatics. My contact details are obviously here, and if you have any questions, feel free to contact me at any point. I hope this helps. Okay, so without further ado, we can begin. So again, these are the lectures that the session is going to be covering. And moving on, we're going to be starting off with some basics just to get into the game. So histology, histo means tissue, and logy is obviously a suffix that's added that means study of. So histology is just the study of tissues, and that's what this entire subject is about. So histology has certain roles, and it's essentially that it completes other disciplines. So what that means is when you're studying anatomy, which is the microscopic picture of things, histology comes in and completes the microscopic picture of things. So you could be studying the liver and its bigger picture, and then you can go down into the details and study what each cell looks like and what it does. It also gives a structure function distribution, which essentially just means it'll tell you the structure of the cell and how it helps its function. So essentially why the organ does what it does and how it does it. And again, it's used in the medical field in terms of histopathology, and you can use it to deduce like tumors and problems and so on and so forth. So now we can get into epithelium. So when we start off with the organization of the body, we have our very basic building block, which is a cell. Then when cells come together, we get our tissues, which is the main focus. And then tissues come together and they give you an organ, and organs come together and they give you your whole organ system and then therefore body. This we know. Then when we talk about tissues, they're organized in a very detailed manner, and it's very purposeful. And this is again to do with their role and the function that they have to perform in the organ that they're in. And tissues have four main types. So you can have nervous tissue, so this includes your nerves, this includes, yeah, that's pretty much it. It includes your nerves. You can have epithelium, and epithelium has many types. You can have muscular tissue, and it's multiple types, and you can have connective tissue. Again, this lecture focuses on epithelium and connective tissue. So the way histology works is like, how do we get into it? How do we get the slides that we have? We have three main steps. So first you have to process the tissue. What that means is essentially you have to cut the tissue into very, very thin slices. You can't take a whole liver and just put it under the microscope and hope things will work. That's not how it works. You have to cut it very, very thinly, and then you have to do certain preservation techniques. So the preservation, before we get into the preservation techniques, we have some angles of cutting, and this becomes important later on in the lecture, where you'll see that the same structure can have like different appearances. So essentially, at this point, like at the top of the intestine, you could have like a circular cut. But if you cut it more at the bottom, you could get like an oval cut. And again, if you cut it more towards the bottom, if you cut it vertically, you can get something that's neither circle nor ovular. So the angle of cutting very, very importantly affects how you view the organ. Then we come into preservation techniques. So there's five in total, and they're very, very simple. So first you have to fixate the tissue. So what you do is you have to inactivate any enzymes that are in the tissue that can cause it to degrade. So you do this by the process of fixation. They haven't given more details on that. And once you've inactivated those enzymes, you're now preserving the structure. So over time, this tissue, it's not going to degrade and rot. Then you have dehydration. So now you have to remove all the water from this tissue, and you want to replace it with alcohol. And then you come into clearing. So now you have all this alcohol, you want to remove it as well. And then you come into infiltration. So you introduce something called paraffin, melted paraffin. It's kind of like a waxy structure. And then after infiltrating, you want it to embed. So you're just going to wait until this paraffin sets. And now you have your slide and it's ready. Then you can stain the slides because they're going to be colorless if you they're going to be colorless on their own, so you want to stain them. And for that, you use hematoxylin and eosin, right? So there's two parts to this dye. Hematoxylin is a basic dye, and so what it does is it attracts anything that's acidic. So it can, say you put this dye, it's going to stain the DNA, which is deoxyribonucleic acid. So you're going to see the color of hematoxylin in the nucleus, and the color of hematoxylin is purple-blue. And then you have, Yeah. And then you have eosin. Eosin stains things pink. And unlike hematoxylin, um, eosin is, sorry, an acidic dye. So it's going to attract to anything that's basic. Okay. And then eosin is found in the cytoplasm. So everything else besides the nucleus is what's going to be stained by eosin. So we have a picture of it here. You can see that the pink part over here is eosin, and then the center part, which is marked by the multiple blue arrows, that's the hematoxylin staining the nucleus. Then once we have tissue preserve, we have processed the tissue by cutting it. We've stained the tissue with certain dyes. Now we come to viewing the tissue itself. So for that, you have microscopy. So you have multiple types of microscopy, but the ones that are main focus for us, especially in uni, are light microscopes and electron microscopes. The light is what you see in labs. Electron microscopes, they're not present here. For your further information, phase contrast is something that you can use on live animals and live structures. And fluorescence is just something that's going to light up. You don't need to know it for your exam for sure. So starting off with light microscope, what light microscope does is it shines light from under. So this is your slide. It's going to shine light from under the slide. And then you have a lens at the top, and the lens is going to take the light that passes through the structure, and it's going to manipulate it and move it in a way where you can now see a proper image. Important points for the light microscope that you actually need to know for your exam are resolution. So what resolution means, it's like the smallest distance between two structures by which they can be seen as separate objects. This definition is very important. So essentially just means like you have two separate objects, but if you view it from like a very further distance, they're going to appear as like what's it called? One small object. But if you like zoom in closer and closer, you'll be able to see that there's two separate objects. So the resolution of a microscope is really important to help you distinguish two cells from each other and so on and so forth. So a light microscope's maximal resolving power is 0.2 micrometers. Important question, this can come as recall. And for resolution, it is a definition, so it can just come as a definition. They're like, which one of these is the definition of resolution, or which one of true or false is this a definition of resolution, and so on and so forth. Then you have electron microscopes. Electron microscope, again, the method isn't the most important, but just for your information, they shine, they don't shine, they like, I guess, throw a beam of electrons towards the tissue, and then it passes through, and again, it's manipulated, and they converge and diverge, and they give you their image. Again, what's important here is the resolution and the maximal resolving power. So you need to know the difference between the electron microscopes and light microscopes resolving power. For electron microscope, it's 3 nanometers, which is even smaller than light microscope. So you can like see even closer up in an electron microscope than a light microscope. Very, very important to know this number and the one previous to it. Okay, so that's our microscopes done, and that is how a histology done. Then we come back into tissues. So we said tissues are a group of cells that come together and they perform a specific function. Tissues have more than just cells. They also have something called an extracellular matrix. So you'll have your cells, and then you'll have everything outside the cells. So this becomes important when we're going to discuss the different types of tissues. There are differences essentially in how much extracellular matrix they have as compared to how many cells they have. So what that means is in nervous tissue, for example, you're just packed with cells. So you have most cells in the nervous tissue, right? And you have very little extracellular matrix. As you move onwards, you'll see that the extracellular matrix consistency increases. So in connective tissue, for example, you have the highest number of extracellular matrix, or not number, highest amount of extracellular matrix, and you have the lowest number of cells. And then these are just intermediate. So they have a similar number of cells to ECM ratio. Like it's pretty basic, it's pretty moderate. So with that done, we will now come into epithelium itself. So what is epithelium? If you can look here, it's essentially some cells that are lying on a basement membrane. Okay, and what they're doing is they're protecting this environment on the inside from the external environment. This is external and this is internal, and it goes deeper. So that's what they're doing. They're separating them, and this is done for protection, for absorption, for a bunch of functions, and we're going to get into them. So there's some exceptions to where you can find epithelium, and besides these exceptions, you'll find them everywhere else. So you will not find them in articular cartilage, so the one that's found for your joints. You won't find epithelium in your tooth enamel. You won't find what's called epithelium in the anterior surface of the iris. Just some exceptions, good to know. Then, um, so yeah, and then we also need to know where epithelium comes from. So it's actually from all the types of germs. Ectoderm, mesoderm, endoderm, it's from all of them. And a good thing about epithelium is it's continuously renewable. So it can constantly regenerate through mitosis. This can also be a question that you can get. It could be true and false, select which one is true. Epithelium cannot regenerate. Epithelium can regenerate. Epithelium can regenerate through mitosis, or epithelium can regenerate through meiosis. So this sentence on its own is so manipulatable for questions. You also need to know that they're polar. So what that means is they have sides. Like you have an apical side, you have a lateral side, you have a basal side. What they mean is, okay, this is an apical side, the one that's facing the outside part. This is the lateral side, which faces the other cells. And then this is the basal side, which faces the basement membrane. So that's what it means. It's polar. Like each side has a role, and it's polar in that sense. And then we come into the function of epithelium. So epithelium has three functions, or three general ones, and then the other ones come under it. So you have protection, you have secretion, and you have absorption. Then excretion can come under, yeah, then excretion comes on its own, and then you have gas exchange, which can come under absorption and secretion. Okay, so again, three main functions or four main functions: protection, secretion, absorption, excretion, and gas exchange. And obviously, excretion, you would see it in the kidneys, so the kidneys epithelium, and the nephrotubules. And then you have the gas exchange, which would be in the lining of the lungs and the alveoli, and protection for your skin, for literally every organ in its capsule, and so on. And secretion, you would see this in the glands, and absorption, you would see this in the um, intestines. We'll get into this for sure. So this is just an overview. So again, protection for literally everything, secretion from glands, absorption from your intestine, that's the most common one, excretion in your kidneys, obviously, and gas exchange in your lungs. Function done. Now we need to characterize the epithelium. So epithelium has three main features that you characterize it with. So the shape of the cell, how many layers we have of the cell, and if there's any special features. And the special features will come a bit afterwards. So when we talk about shape, there's three main shapes that you could have. You could have squamous, so they're very flat and they're very thin. So if you come here, these this over here, this is the nucleus. So this entire thing is a cell. So it's very, very thin, very flat, and they're very easy to tell. This again is a cell, this is a cell. So again, squamous cells, very, very thin, very, very flat. Then you can have cuboidal. So cuboidal, as the name suggests, they're cube shaped and they're very, very clear in this image. It's like a cube, right? Then you have columnar. Columnar, again, column, pillar. So they look like a pillar and they're very long. And yeah, and these shapes again also have contributions to what function they're performing. Squamous, because they're very, very thin, these are very good for diffusion because for diffusion you want very little distance. Cuboidal and columnar, they're really, really good for secretion and absorption. And we'll come into it because again, they contain more space for more features that can help with their functions. Okay, then we have layers. Now you can have one singular layer or you could have multiple layers, right? So here, for example, so you don't see another cell like this under this, right? There's nothing here. And this is not this. So you just have one layer of these cuboidal cells, and under them you have the basement membrane. So this is one layer. Then you have stratified. So you can see that there is multiple cells, multiple nuclei here, here, here. They're all the way here. And sometimes with stratified cells, it can get confusing to figure out what type of a shape it is. So like if you were zooming in here, you would think, like in this section, you would think it's cuboidal. But the actual way to read for stratified cells, you have to come at the very, very top. So you can see at the very, very top, the cells are flat and thin. So this is actually stratified squamous. So that's the way you read stratified cells. You have to come at the very, very top to be able to tell what type of cell it is, because in stratified they're layering upon each other and the shape looks different. So now we come into naming. So you would say layer, cell name, and then epithelium. So this we said that these are squamous cells. You don't see any other layer under this. They're attached in a very prominent basement membrane, this pink line. So this is simple squamous epithelium. And then we have this one, cuboidal cells, only one layer, simple cuboidal epithelium. Don't confuse this part, by the way, for this part. These are two different sections. Now, as I mentioned, because cuboidal and columnar cells, they're a bit larger and they're a bit bigger, they have more space to like accumulate mitochondria, and this helps with their function, which is secretion and absorption. Here we have our simple columnar epithelium. Then we come into the special features. So special features, and there's also in more detail later, but this is just for naming. You can have little finger-like structures at the top, and there's a large variety of them, and they're called cilia. Cilia's function is essentially they move back and forth and they can transport things. So you'll usually find this in your respiratory tract, and they take the mucus away from your respiratory tract and they move it all the way up so it can go down into your digestive tract instead, so it's gone. Then you have keratin. Now, keratin is a type of connective tissue, and it's this thing. So here is where our cell ends. And I want you to pause the video and think about what type of epithelium this is. And then I'm going to tell you right now, it's also squamous epithelium. It's a very, very flat layer at the top, and it's squamous epithelium, stratified squamous, because there's multiple layers. And then you have this point at the top, and it's wavy and it's pink, and this is keratin. All right, so for this, you would say keratinized stratified squamous epithelium. You also have it in this picture, but it's not very clear. And again, here you can see all the cilia. They're very, very prominent, very easy to tell. And yeah, that's stratified, that's the special features. And then we have some special cases. So these are the ones that don't fall into any of the rules that we gave for the naming. So these cells, if you look at it from the top, you would see that there's like nuclei upon nuclei. So like you would think there's two layers, like you would think this is one layer and this is two layer. So you would think that there's two layers, but there's only actually one layer. And so it's called pseudo stratified. Pseudo means fake. Stratified means that it looks like it's stratified. So pseudo stratified. And okay, the way you can tell is each cell, literally all of them, they're attached to the basement membrane. And there's only one that's attached, and they're like all of them are attached to the basement membrane, but not all of them reach to the top. All right, so whatever we thought was the second layer, like say this, we thought this was a second layer, it's actually attached to the basement membrane, so it wouldn't be a second layer. A second layer would be sitting upon a pre-existing cell. I hope that makes sense. And again, there's our cilia, the special features. Now for the histology image of it, again, it looks very messy. It looks like this one definitely looks like it could be multiple layers, but if you like trace it down here, it's all coming to the same basement membrane. Ooh. And again, it has a lot of cilia. So pseudostratified columnar epithelium. Yeah, and these are mostly found in your respiratory tract. Then we have something called transitional epithelium. So what these have is they have a special ability to essentially change their shape very slightly. So they can be very, very flat like this, and then they can start expanding. So they look like, so they're just transitional. They can change their shape. They have the ability to expand or I guess shrink back to normal. You'd find this in the bladder. So when our bladder is sitting normally, all the cells are in their normal shapes and sizes. But so let's say this is our bladder and this is in normal capacity, it's not full. So all the cells are like sitting normally in here. Then say that the bladder is really full and the bladder expands, right? You won't have to pee immediately after you drink water, so the bladder can expand. So what happens is these cells, they like expand like this, like they become flatter and thinner to make the bladder bigger or more voluminous. So that's what transitional epithelium is. So again, they can distend, they can expand. And this here, for sure, you can tell that these are stratified because the multiple layers are sitting upon each cell instead of all of them sitting on the basement membrane. Now this over here is just a summary of all of the types of epithelium we can have. Simple squamous, simple cuboidal, simple columnar, then stratified squamous, stratified cuboidal, stratified columnar, keratinized, non-keratinized, ciliated, so on and so forth, and transitional, non-transitional. That's done with the types of epithelium that we have. Then we have the basement membrane, the main guy that we're talking about, on which the cells sit. They're very, very clear here. You can see them. It's like a filter. So it's very, very, it's like a filter, and what it does, it's very selective on the things that it can let like pass through. So it's semi-permeable. It'll let certain nutrients pass through and certain things cannot pass through. That's just our basic understanding of things. Now, basement membranes under an electron microscope actually have two layers. So they have a fibrous reticular layer and they have a basal lamina. So the basal lamina is the one that's facing the cells. So let's say we have two layers. This is the epithelial cells. This is going to be the basal lamina. And then the one on the bottom is going to be the reticular lamina. Okay, and now there's some features about them that you definitely have to know. These can come very easily as a recall question because Dr. Khalid doesn't very frequently give like a lot of histological pictures in the MCQs. He can give a few, but most of the times it's going to be like recall questions of things like these. Very simple though, not a lot of critical thinking, but just recall. So you need to know which type of collagen fibrous reticular lamina is linked to. So the one it's linked to is type 4 collagen. And then the basal lamina, it's linked to type 3 and type 7 collagen. You need to know them. And then these names, not very, very high yield, but good to know that they're linked to fibrous reticular lamina as opposed to basal lamina. So again, collagen, type 4 for fibrous reticular, type 3 and type 7 for basal lamina. Okay? You could do like 4 plus 3 is equal to 7. And then this is basal, and this is fibrous, fibrous reticular. But that's just if it's that hard. But yeah, this should help you, inshallah. Then we come into cell junctions. So how are these cells, like, you want to wonder, how are these cells sitting together? How are they sitting like next to each other and together on the basement membrane? But like they could very easily move and be dispersed. So you have little junctions that help like anchor them to their place as well as keep them like in communication for where it's needed. So you have three main types of cell junctions. You have tight junctions or occluding junctions. Good to know both names because they help in the later part. There's also anchoring junctions, and then there's gap junctions. So with tight junctions, as the name suggests, they're very, very tight. They don't let communication go between cells. So this is really important for um, certain epithelia where you don't want things to pass through. So it's just going to maintain a very, very like strong line. It's not going to be like break and then like this. It's not going to be very permeable. So what's it called? Epithelium that have tight junctions, they're not very permeable. They're very, very tightly packed. And what's it called? And they're usually between like cells. So they could be between this cell and this cell because you don't need communication between these two cells. So you have tight junctions. And what it has three contributing proteins. So it has occludin. That's where the name occluding junctions, to remember with tight, is really helpful. Occluding junctions, so occludin is part of it. Then claudins. Once you remember occludin, then you can remember the other name that sounds similar to occludins, which is claudins, is with it. And then junctional adhesion molecule, so jam. Think of it like jam is quite sticky sometimes. Sometimes it can be very sticky, so it's helpful in like gluing things together. So that's what tight junction is. It's like a glue holding the cells together. Now Dr. Khalid doesn't always bring clinical stuff. He actually doesn't. He mostly tells us that he's not going to bring it, and he never does. But just for your information, good to know just in case. There's a bacteria, H. pylori. It can release a toxin that disrupts the tight junctions between the stomach lining. So they could tell you, worst case scenario, you get a question about a person having symptoms of like stomach pain or so on and so forth, and they find out that he has H. pylori. What happened to his stomach? So tight junctions were disrupted by toxins released from H. pylori. Then you have anchoring junctions. So we're the second type. Anchoring junctions has two subtypes. So it has junctions that are between the cell and the cell, so between these, and then they have between the cell and the environment. So here and yeah, between the cell and the environment. So these are the two types of junctions you have. So cell to cell, you have desmosomes, and these are for intermediate filaments. It is good to know the type of filament that is associated with the junction, just because sometimes you can get questions on them. So desmosomes, very, very strong, and they're from intermediate filaments, which are kind of the main filaments. So desmosomes. Then you have adherence junctions. So these are like, so say we have our tight junction here, they're below the tight junction. Can you see this? Yeah. Tight junction here, they're usually found below the tight junction. Um, you don't need to know the whole process of it, but just to like remember some keywords, right? So adherence is related with coadherence or C adherence, whatever we want to call it. Sounds very similar, so they're together. And C adherence is related to catenins. Again, C ins, very similar names that go together. And they do this in the presence of calcium. So C, and C, three Cs, they go with adherence junctions and they're cell to cell. And these are linked to actin filaments. So remember A, actin filaments, adherens, cadherins, catenins, calcium together. Just remember them like this. So again, desmosomes, intermediate filaments, strong attachment, adherens, below the tight junction, links actin filaments together using C adherens, catenins, and calcium. Then we have cell-cellular environment, so cell to the environment. So you have something called focal adhesions. So they connect the actin cytoskeleton to the extracellular matrix. So we said we have cells and then we have everything outside it. So what's connecting you to it? The focal adhesions. And then with focal adhesions comes these three names. Not most high yield, I would say, but good to remember anyway. Paxillin, vinculin, and talin. Their names are very, very similar. So if you remember these three together, then you can just link them a bit more easily to focal adhesions. Then we have hemidesmosomes. So what hemidesmosomes do is they connect the epithelial cell to their basement membrane. So it's a connection between these guys over here. Important point to note, we have had a question about this, and they're really important for like epithelia where like you could face abrasion. So in your skin, like your skin can get damaged. So it's good for that. For the mucosa of the oral cavity, where eating this layer constantly is subjected to wear and tear. Same for our cornea, very, very delicate part. And hemidesmosomes, it's composed of plectin, BP230, and urban. Um, I don't really have anything for you to memorize these, so keep them like low priority, and if you have time, just go over these again. Then we have the last type of junctions, which are gap junctions. So gap junctions, as the name suggests, they have a gap in them. So these are the type of junctions that you actually use for communication. So the previous junctions, they're only for connecting. Tight junctions don't let anything go through, and the other junctions are just for connecting. So how do you communicate between cells? Now, this is important for cells where permeability affects like the action potentials they're doing or the movement of fluids and so on. You'll learn them later as well, and you'll see specific systems where gap junctions play a very important role in the epithelium. But for us, all we need to know is gap junctions. They're maintaining communication between the channels, between the cells. Gap junctions are composed of connexins. So 6 connexins come together and they form a connexon. So it's like 1, 2, 3, 4, 5, 6. These are the connexins. Individual ones are connexins. This entire thing is a connexon. They come together. You have the gap junction, which is what these are. This is how they look from the outside, and this is how they look when you cut them. So that's gap junctions for us done. And then we're going to come back to the special features that we were discussing. We have 3 special features that cells can have. So we can have microvilli. Now these can look similar to cilia, but what you need to know is that they're shorter than cilia. So you can see cilia are very, very long. Microvilli are very short. They're truly very, very short. And their purpose is to just increase the surface area of absorption. And these are usually found in the intestines. Now, what they're made of, also very important, we have had a question on this. They're made of actin and microfilaments. So actin and its relation to microfilaments, those come together for microvilli. Then you have cilia. As I mentioned, cilia help move things back and forth. There's 3 types of cilia. There's motile, primary, and nodal. You get a question on this and it'd be like, what are the 3 types of cilia? And they could say in one option, motile, primary, nodal, motile, secondary, primary, so on and so forth. So it's just good to know that these are the 3 types. Cilia are also associated with these names. So basal bodies. So what basal bodies is essentially, you see the cilia and then you see this dark like outline on which the cilia are sitting. This dark outline is created by something called basal bodies. What you need to know is that basal bodies is linked to cilia. Then there's also how they're formed. So there's a 9+2 assembly of microtubules, which make an axoneme, and I'll get into this in a bit more detail just for your understanding. Then you have stereocilia. So they're long and they look like cilia, but they're in very less amount. And these are usually found in what's it called, the male reproductive system, for example. Ooh, stereocilia. And now, as the name stereocilia suggests, they don't move. They don't move back and forth. So what they do essentially is they just increase the surface area of absorption. And these are also like microvilli made of actin and microfilaments. So you can link it together by function. Both increase the surface area of absorption, so they're both made of similar things. And then the other one is motile, it's moving things, so it's going to be made of something different. So here we come into the axoneme. So essentially, this entire thing is called an axoneme, and it's making like a cilia, one singular one. And you have a 9+2 arrangement. So what this means is this is 1, this is 2, this is a 2. This is a 2 of the 9+2, so we're done with this. Then you have 1, 2, 3, 4, 5, 6, 7, 8, 9. These 9, these pairs, they are the ones of they're the part of 9, I guess, 9+2. And they have little subunits A and B, and they share them and they don't, and there's a whole thing. Again, not very, very important. More important to know the 9+2 arrangement, there's 9 pairs of microtubules are on the outside, and there's 2 single ones in the inside. Important to also know pairs and single. So these are 2 single microtubules, but these are pairs, but they're still counted as like one pair. They're the one pair of the 9. Then they're also connected by proteins like dynein and nexins. These are what help them move. So these are the things that are providing them the ability to move back and forth, which is what's contributing to their function. So that's ciliary axoneme done. Then we come into glandular epithelium. So as I mentioned, we have cuboidal and columnar cells, and these are more involved in glands. So essentially, these cells are really specially characterized, for secretion. So they're going to release things. So some embryology background into it is essentially these cells are growing, and then, okay, so you have your epithelial cells here, and under you have a layer of connective tissue, and they're growing. And what the epithelial cells do is they start to penetrate inside. So they go into the layer of the connective tissue, and they go deeper and deeper, and they can form 2 types of things. They can either form like a duct system and then a whole gland, or they can just form a gland and then this connection can disappear. So again, they penetrate into the epithelium, into the connective tissue, and they go deeper. They can form a whole gland or a gland there. And then if they maintain the connection like this, they have a duct. So they're exocrine glands. They don't maintain this connection, so it's gone. It's a ductless gland, so it's endocrine gland. And yeah, that's their entire importance essentially. So now our main, sorry, before I get into that, our the difference between exocrine and endocrine glands, besides the duct, is obviously how they release things. So that again comes into that same feature. Exocrine glands, because they have the duct, they'll just produce their materials in the secretory portion, and they're going to release it through the duct into the surrounding area. Endocrine glands, because they don't have that feature, they'll mostly release it in the bloodstream, which should be near the glands, essentially. It's going to release it in the bloodstream, as you can see here. And it can also do something called paracrine, which is where they release it to the nearby cells and it affects them. The main focus for this lecture is an exocrine gland. So it has 2 main features. It has the duct, as we mentioned, and then it has the secretory portion, the part that's making the secretion of the gland, and that's going to be called the acinus. Now, this portion, the duct stays the same. It's just duct, but the secretory portion, the acinus, it can have multiple like morphologies. So you could have unicellular. So this is a unicellular gland. You can see it's just one big lump. Yeah. Sorry, just a second. It's just one big lump. And then, yeah, moving on. Then you have the multicellular secretory portion where you can see that there's multiple lumps right around here. So that's just it. Unicellular like this, or multicellular where there's like many nodules or many I guess, yeah, collections, and they're all surrounded by one lining like this. And then these are the different types of it. good to know what they look like. I'm not really going to get into the detail of it because that's just like the names are very, very clear and their morphologies are clear. Then you have some mechanisms of secretion. So how are exocrine glands going to release them? So what you can have is you can have merocrine secretion, which is what happens is you have a cell and you have little vesicles of the secretory material that they're producing. The vesicle come and they fuse into the lining, and then the materials are released outside. So that's one method. So this is merocrine, vesicle inside, and they release their materials through exocytosis. And you can see this in pancreatic acinar cells. Then you have apocrine, which is you have the cell and it's releasing the secretions in like these are the secretions, the little dots inside the circles. So you have your secretions and they're surrounded by a layer of plasma membrane. So they're enveloped. So they're like little packages of what's it called, secretions. So you'd see this in lactating mammary glands, moles glands, and external auditory meatus glands. Just good to know the examples. They can just give the name and say so just what type of which of the following is the type of secretion method they follow. Then you have holocrine, which is essentially like okay, you have the cell and the secretions like they pile up inside the cell. So this is a nucleus and everything piles up inside the cell. And then the cell undergoes programmed cell death. So the cell bursts and all of its contents are released outside. This is seen in sebaceous glands and tarsal or meibomian glands. Here is a histological picture of what they look like. So these secretions can be serous or mucus. So this is serous and this is mucus. The difference is that serous are not glycosylated and mucus are that they are glycosylated. So they contain mucins. So the mucins mucins are something mucins are a type of secretion you would see in goblet cell secretion. So this is the secretion that you see for your mucus, which is lining the respiratory tract. Then you have serous. This is the one that you can find in some of your salivary glands. Sorry. And yeah, so the difference is the serous look very clear, right? And the mucus look very pink, very packed. Yeah. Oh, yeah. And that is pretty much it for epithelium. That is all of epithelium covered. So I have some questions for you. Just pause and think of the answers, and then I'm going to go into the answer. These are all recall questions, mostly, because that is the type of questions that he usually asks. Some of them might have multiple answers. So yeah, just answer when you answer after this. So for this question, um, yeah. So for this question, oh, sorry, and D. Okay, so for the first question, which of the following can be used to differentiate between types of epithelium? So number of layers and shape, number of nuclei, position, cytoplasmic content. Obviously, you would use number of layers and shape, and this was a question that we did get. Then, which of the following types of connective tissue consists of one layer of cells? Sorry, which of the type of epithelial tissue? Sorry for the typo, consists of one layer of cells. So simple columnar, stratified squamous, transitional, or pseudostratified columnar. So for this, the answer is A and B. A and D, sorry. Yeah. And that is it for epithelium, so we're done with that. Now we're going to come into connective tissue. So remember our scale of increasing to decreasing ECM. So connective tissue sits very high on at the very end of this. It has the highest extracellular matrix. So yeah, so this is what connective tissue can look like. So you'd see that these very like little dots, these are the types of cells that you would have in connective tissue, very, very little. And the rest of this red jelly that's surrounding it, that's extracellular matrix. That is what connective tissue is like. It's just very few cells and a lot of ECM. So the ECM is made of something called ground substance, and it's also made of protein fibers. And we're going to get into what those are. Then we talk about the function of connective tissue. So connective tissue essentially provides support. It binds things together. It can store energy, and it also helps in transport. And it can also be used in repair. So yeah. So these are just some examples of what connective tissue look like. If you remember the keratinized epithelium that we were talking about, this entire thing is connective tissue. Now, okay, we talked about this. This is our epithelium, and they're sitting on the basement membrane. Everything under the epithelium, this whole pink swishy stuff, this is connective tissue as well. So there's many types to it. And you have that, and then you also have this entire image over here. All of it is connective tissue. And you can see that there's very few cells and then everything else around it. This is just a schematic diagram. We're going to get into it. So connective tissue has multiple types. So you have embryonic connective tissue. So this is the one found in fetuses. So you can have mesenchymal or mucus. And we're going to get into pictures of these as well. So mesenchymal, these are the ones found in the embryo. Important distinction to make it from mucus connective tissue, which is in the umbilical cord. So you can find mucus connective tissue in the embryo, but you cannot find mesenchymal connective tissue in like a newborn baby and in their umbilical cord essentially. And the mucus connective tissue is rich in ground substance. Okay? And it has an appearance of Wharton's jelly, and we're going to get into that as well. Then you have adult connective tissue. So this is the one found in like our bodies, and it has types. So it has loose connective tissue, so loose areolar connective tissue, or it could have dense connective tissue. So as the name suggests, it's on the packing of this, like how densely packed is everything versus how loosely packed it is. Now, dense connective tissue can have two subgroups, dense regular versus dense irregular. In dense regular, they're arranged in a very like distinct pattern versus dense irregular, where they're in random movements. Then you can have another category called reticular connective tissue or elastic connective tissue. Then you have some special connective tissues, and these are essentially your adipose tissue, so the white and brown adipose tissue. They're also your cartilage, and they're also your bone and bone marrow. Cartilage has three types: highly inelastic fibrocartilage, good to know. Bone and bone marrow. The types of bone you have are compact or cancellous. Compact, obviously, the outer part; cancellous, the part that's more porous. And yeah. So, connective tissue, we need to talk about its composition before we move into the morphology of it. So, connective tissue cells. Now, you can have two groups of cells. You can have resident cells, so they stay in their location, and you can have wandering cells, which move around. So for resident cells, you have fibroblasts. So remember that the suffix blast is for immature cells. It's good to know. So fibroblasts, they are the producers of the extracellular matrix. So they produce the collagen, the elastin, and the ground substance. So these cells stay where they are. If this is the connective tissue of your skin, then these fibroblasts will stay on the skin area. They're not going to go down deeper into the organs. Adipocytes, these are also, they're going to stay wherever they were forming. They're not going to move around. They're not going to travel in your bloodstream and go to different places. They're going to stay in their original location. Then you have mast cells. Mast cells are involved for immune responses. We'll get into it. And then you have macrophages. Now, macrophages are in both and technically kind of in both. And these are resident cells. They'll stay in the location of the connective tissue they were assigned to. And then you have plasma cells. So these are the ones that are producing antibodies. Again, these will all stay in their location. For wandering cells, literally all the other WBCs, the monocytes, the basophils, the eosinophils, the neutrophils, they'll all be wandering cells. So they travel around, they go to the site of infection if needed, or the site of inflammation, they carry out their function, and then they undergo apoptosis. They're not long-lived, as opposed to macrophages, which are long-lived. Important distinction to note that monocytes, from which macrophages are derived, monocytes are wandering. So they'll go to the site of infection and then they'll stay there, right? Where they become macrophages, and then they're resident. So if we go down one by one, fibroblasts, so their main function is, again, they make the entire extracellular matrix, right? So they're maintaining it and they're synthesizing in it. Then you have the structure. So there's two types of fibroblasts. They can be active or they could be quiescent or quiescent. Quiescent just means they're not very active. So think of it like quiet. So active fibroblasts, okay, the way to note them is, okay, these are active fibroblasts versus these which are not active. Okay, so active fibroblasts, they're long, they're elongated. So this guy over here, and they have euchromatic nucleus, which means it's a loose chromatin. They have a lot of rough endoplasmic reticulum, and they have a well-developed Golgi apparatus. So these are found near collagen bundles. So these are also active fibroblasts. Then you have quiescent fibroblasts, or the tiny ones. They're smaller. They don't have that much rough endoplasmic reticulum, and they contain, excuse me, a heterochromatic nucleus. So that means it's tightly packed. So the smaller cells, right? You can see the difference between them. So once quiescent cells become active, then you would have that appearance. Then you have mast cells. So mast cells are cells that are really important for allergy and inflammation. They contain multiple so what's it called, hormones, so histamine, heparin, and also chemotactic factors. Um, so histamine is something that's always linked to allergy. So you think mast cells, you need to think histamine, you need to think allergies, right? That's how your thinking process should go. Histamine increases vascular permeability, so it makes the vessel more permeable, and it also contracts smooth muscles. So this can cause some vasoconstriction as well. Heparin, it's an anticoagulant, so it's gonna, as the name suggests, anticoagulant. You guys are doing HLS, so you would know better. And then chemotactic factors. So they have things. So once this mast cell, what it does essentially is it'll release all of its contents during an allergic reaction or an inflammation or something going on. And so once it releases the chemotactic factors, these are like little signals for the WBCs to come to the site of where the mast cells went haywire. Then you have macrophages. So macrophages have the main function. Sorry, give 1 second. So yes, okay, macrophages have the main function of phagocytosis. So they're gonna clear the environment of pathogens, that cells, and so on. They come from monocytes. Good to remember. Remember, macrophages are resident and monocytes are wandering. They are involved in inflammation, so they come to the site of infection and they clear it up. Very, very important for it. And then good to know some names. So macrophages, when they go into their distinctive places, they have a different name. So in the nervous system, they're called microglia. In the bone, they're called osteoclasts. And in the liver, they're called Kupffer cells. And if you look closely, you can see that so these are the macrophages, and their nucleus, it looks kidney-shaped. So I guess the best example would be this guy over here. Kidney-shaped nucleus with a nucleolus in the center as well. Then you have plasma cells. These are the ones that are producing antibodies. They have a clock face appearance. So if you look closely, it kind of looks like a clock, you know? So that's what plasma cells look like. They contain both heterochromatin and euchromatin, and these are again involved in immunity. So that's the cells of connective tissue done. Then we come into ground substance. We said that the extracellular matrix is made of ground substance. So ground substance has 3 main contributory factors. So you have something called glycosaminoglycans, or GAGs. So they're polysaccharides, and they're made of repeating disaccharide units. Too much hassle to actually think about it, so just memorize the names that are related to it. So glucosamine, galactosamine, and glucuronic acid. They sound very similar to the GAG name, the glycosaminoglycans. Try to link it together from that. What their purpose is, so they're a type of sugar, right? So what they're gonna do is it's gonna attract water to wherever they are. This brings in the water for the ground substance, and it helps it have that appearance of the jelly-like consistency. So when it forms this type of texture, it's a really good shock absorber. So yeah, so think of it like, where is it? Our jelly was right here. So this was like, if you hit the jelly, not too hardly, it's gonna bounce back, shock absorber. It's not gonna let the cells around it get damaged. That's what I mean by shock absorber. Oh, we forgot to cover adipocytes. So let's get back to this as well before I finish that ground substance. Adipocytes, there's white and brown, as we mentioned. The brown is what you see in newborns, and the white is what we all have. They're obviously different in appearance, and also they look very, very clear. And this goes into the process of tissue preservation, where you clear it with water and you clear it with alcohol. That's what causes it to look white. It's cleared out completely. Anyway, moving back to the glycosaminoglycans, you have some types, and so you need to remember these names. These can come in your exam as well. So hyaluronic acid, this is the most abundant one, very important, easily recall. Then you have chondroitin sulfate, dermatan sulfate, heparin sulfate, and keratin sulfate. Their names actually align with where they are. So chondroitin is for the bones, dermatan, they are near the skin, and so on. Then, after glycosaminoglycans, you have proteoglycans. So these proteo, the name suggests it's a protein, and then they have glycans, so they have the GAGs attached to them. So if we come to this picture here, and this is a proteoglycan. So this is your protein, right over here. This is so this green part over here. Then these little dotted lines that are attached to the green line, that's your GAGs. So these can be hyaluronic acid, these can be dermatan sulfate, and so on and so forth. Then also important to note that these are synthesized in the rough endoplasmic reticulum and they mature in the Golgi apparatus. Not the most likely question to come, but it can come. Examples of proteoglycans can be agrican, versican, perlecan, decorin. The name sounds similar to proteoglycan, so you should be able to piece that together. Yeah. Okay, and 1 second. Okay, and then the last two contributing factors that we have multi-adhesive glycoproteins. So these are proteins and they have binding sites for cells, collagen, proteoglycan. Just to know the general idea of it, what you need to know are the examples. So fibronectin and laminin, these are the two examples that you should know, and these are in your slides. So fibronectin binds to collagen, proteoglycans, and integrins. So 3. And then you have laminin, which binds to integrins, and type 4 collagen. So these two could be linked together quite easily. And yeah, and then you have the last component, which is water. So water in the ground substance and in connective tissue language, it's actually referred to as interstitial fluid. And it's a solvent for nutrients, so for the oxygen, for the everything else, every other nutrient that you have. Then, if you have an accumulation of interstitial fluid, if there's too much of it, you're gonna have edema. Again, a clinical point that can come in a question. Okay, then we're talking about how is connective tissue degraded. So you have enzymes called metalloproteinases, or specifically matrix metalloproteinases. These come and they help in like changing the shape of the ECM, changing its components, and degrading it if needed. And so matrix metalloproteinases are inactive proenzymes and they're activated when they're in the extracellular matrix. Important point to note for sure. And then we come into collagen. So collagen is, as we mentioned, a type of connective tissue, and it has like a bunch of types. So you can group them in multiple ways. So this is just the types mentioned, and this is grouping them based on certain functions. This is a very important slide. This is very easily recall questions that can come from any of this. So type 1 collagen, this is the one found in skin, bones, tendons. Type 2 is found in cartilage. Type 3, this is very, very important. It's mentioned again and again. It's found in reticular fibers. You have type 4, which binds to the basal lamina, also important. Type 5 for placenta, and type 7 for anchoring fibrils of basal lamina. Then you can also name them according to their function. So fibril forming or fibrillar, that's type 1. So that's as the name suggests. Then you have network forming or sheet forming. They form a sheet of things. So that's type 4, basal lamina, the one also found in basal lamina. Then you have fibril associated. So that's type 12 and type 1, and these are the ones you see in tendons. So yeah. And then lastly, you have transmembrane collagens, which are found in type 18, important for the reticular lamina, which is what we saw in the basement membrane, the 2 layers of it. So yeah, these are the types of collagens that we have. Then we want to talk about how they're formed. So how collagen is formed. So you start off with something called procollagen. So procollagen is formed when you have 3 alpha chains. So you have 3 alpha chains and they come together and they form a triple helix. Ignore how poorly the diagram looks. And this is your procollagen over here, the three alpha chains coming together. Then this enzyme acts on procollagen. It's called procollagen peptidase. And this converts it into something called tropocollagen. And it does this by removing an end, as the name peptidase suggests. Then tropocollagen, so you have this is one. You have multiple of these together, times 3, times 4, however many, and they come together. So you have tropocollagen 1, tropocollagen 2, 3, and they come together and they start forming something called collagen fibrils. And then the fibrils come together and you have your collagen. That's essentially the process. And this is the histological picture to show you the helixes and the triple helix and the procollagen and triple collagen together. So yeah. And then now we're coming into the images, the histological images. So remember there's the embryonic connective tissue, so there's mesenchymal and mucus. Mesenchymal is present in the embryo and the umbilical cord, and it also has gap junctions. Very important to note. Collagen fibers, you can't see them in mesenchymal. Then you have mucus embryonic, which is only seen in the umbilical cord. Very important distinction to make again. This appearance, the way this looks, it's called Wharton's jelly. That's what it was about. Then we come into adult connective tissue. So there's dense regular and dense irregular. So you can look at the picture and you can tell that this is very dense. It's so pink, like it's completely packed. All of this is connective tissue. And even here, all of this entire thing is connective tissue. So essentially, what happens is for dense connective tissue, you have type 1 collagen, and they're very tightly packed together. And you see these little dark purple things? These are your fibroblasts. And now you can see the big, like the vast difference between the ratio of fibroblasts to ECM. So these purple things are your fibroblasts, and again, they're all tightly packed together. Now, because this is so tightly packed, you can imagine how strong this would be. So it's good for tensile strength. And this is found in tendons, so muscle tendons, very, very important. They're found in ligaments and they're found in aponeurosis. These are, again, things that are attaching muscles to bone and bone to bone and so on. Then you have dense irregular. So again, it's very dense. It's completely pink, but it's not linear. Like here, you can see these are like clear lines or columns, even here. Dense irregular, it's kind of flowy, but it's very packed, but it's kind of flowy. So this can resist stress from all directions. That's the function of it. And so you can find this in the dermis of the skin. You can find this in the capsules of the organs. So like organs that have capsules, you have your spleen, you have your kidney. So that capsule is made by dense irregular tissue. And yeah, so remember when I was telling you about how cutting the tissue for tissue processing, cutting it in different angles can produce different images. So here, look at the difference between the cross section and the longitudinal section. Like they look completely different, but they are the same thing. Then we come into loose areolar tissue. So loose areolar tissue, they're not densely packed, and they're not dense, they're not packed, it's just loose. So you can see like, okay, this is your epithelium, and this is your epithelium, and then everything besides the epithelium, that's your loose connective tissue. So it's always found under epithelium. So I'm not in this picture, but I'll go back later and I'll show you under the epithelium, everything that's under the epithelium's basement membrane, it's all loose areolar connective tissue. So you would find a large amount of ground substance. You'll see that they have fibroblasts as well, obviously. They can also have macrophages and mast cells. So what they do is their main function is to support the epithelial tissues. They're under it. And they allow diffusion of nutrients. Because they're very loose, they're not densely packed, nutrients can flow through very easily. And they're also involved in immune defenses, as we mentioned, because they contain macrophages and mast cells. So this is a summary of how they look. So coming from loose to dense, you can see just how greatly like the number matters, like the number of fibers that you have, as well as how they're packed. So here there's very little. Here you can see some coming in together. This red stuff is the connective tissue fibers. Here you can see that it's coming together a lot, so it's increasing in density, but it's not very packed yet, like regularly. And then here you have a tendon sheath, very, very tightly packed. And yeah. Then we come into the other types of connective tissue. So you have reticular tissue and elastic. So starting off with reticular, remember we mentioned collagen type 3. Very, very important. Again, just knowing this link is important. So what reticular fibers do, you need to have a special stain to see them. So this is actually a silver stain, and it makes it look black, right? So um, these guys over here, as the arrows point to, are the reticular fibers, and they form like a mesh, like a network. Um, and yeah, and this is good for like supporting organs in some places. And an important point to note is that they are vascular, so they do have a blood supply coming to them. Very important to note. Then you have elastic tissue. So elastic tissue, you would get probably this image. There's not a lot of images they would bring in the exam if they do bring it. So this is elastic connective tissue. The things that are painted black, that's the fibers. And you can have two main components for them, elastin and fibrillin. You don't need to get into the details of them, just know the names. Um, so an important feature that they did mention is elastic fibers can stretch up to 150% of their length, and then they can come back to normal, so they won't be damaged. This is a good feature. It's good for their function. This is important, say, in your lungs, which need to expand and come back to their normal size. Then we talk about connective tissue and its role in wound healing and also in regeneration and repair. So in wound healing, you have something called myofibroblasts. So you have fibroblasts, which are from connective tissue, and you have myo next to them. So myo, the prefix always means something related with muscles. So myofibroblasts, they are fibroblasts, but they have features of muscle cells, smooth muscle cells specifically. They can contract like smooth muscles can, and this helps in like repairing the wound. So you have a wound like this, and it's open. Once you have the myofibrils, they'll contract and they'll make it like come together and close up. So that's wound healing. Then you have regeneration and repair. So most of the time, they can just replace the damaged tissue and they can return to normal. So say you had like a break in the break, say you had a break in the lining or something as such, it can come in together and it can repair as much as it can. And this is done through a process of regeneration. If regeneration is not enough to repair the damage, then you have a scar forming. So yeah, that's the connective tissue again, but it is forming a scar. Yeah. Then last point of connective tissue is aging. So, essentially, the reason our face starts to look different, our hands start to look different, is because these are heavily reliant on connective tissue as we age, is because connective tissue is decreasing. So, um, essentially, what happens is as you get older, your type 1 collagen, its production starts to decrease, and on top of that, you have excessive degradation. So you're not having that much production either, and on top of that, you're having more breakdown than normal of the connective tissue layer of the skin for the dermis. So decrease in production and increase in degradation, it's going to lead to the changes that you see. And there's also a decrease in the number of elastic fibers. So that's how our cheeks or our hands even, they start to sag and so on. Decrease in the number of elastic fibers, the ones that can expand and come back to normal, as opposed to the thick and non-functional ones, which just stay as is. So that is connective tissue done. I hope that made sense. Again, if you have any questions, just message me and I'll try my best to explain. So these are some questions. You know the drill. I'm gonna go to the answers now. So which of the following is a wandering cell? Obviously all of these, ignore this. This shouldn't be here. These two are resident cells, and this is your wandering cell. I would say to ignore lymphocytes because these do come in wandering cells, but this is a very general answer, so ignore it. Wow, you cannot read that handwriting at all. Anyway, your answer is monocytes. Which connective tissue is found in the umbilical cord? Only in the umbilical cord, it's mucus. One that is found in the umbilical cord and the embryo, that is the mesenchymal. Important to know. Then, which glandular secretion mechanism undergoes programmed cell death? And which type of cell signaling occurs when cells send signals to themselves? I didn't exactly go into this, but you would have an idea because I mentioned one example of it. Anyway, for this you have holocrine, so the cell packs up with the secretory molecules and it releases its contents. And then you have so I mentioned paracrine, which is they release it to nearby cells. Then autocrine, they release it to themselves. Yeah. Last part, which of the following are extracellular matrix components, but not part of the ground substance? And the next, what are reticular fibers made from? So yeah, for the question here, this question was this was a question in our final, but it was quite confusing and no one ended up having the right answer to it. So ignore it. This was just here for fun. Yeah. And this is to give you an example of what you can get in the exam. But yeah, it's a very messy question, so it's one that would be appealed. Then you have what are reticular fibers made from? So that's type 3 collagen, very, very important to note. Hyaluronin is something that's hyaluronic acid is what you see in GAGs. And yeah, I'll remove this question from the final slide, by the way. Okay. And then we come to the last part, which is lymphatics. So this system is made up of cells, tissues, and organs that contain immune cells. So that's what the lymphatic system is made of. So what is its function? So essentially you have your normal blood vessels and then fluid can leak out of them and they go into the surrounding area and lymphatics essentially take it from the surrounding area. So these are vessels that drain the I guess extracellular matrix of the interstitial fluid so it doesn't pack up. So it drains it. It can also carry out immune responses because it does contain the immune cells, so they can come to the site of infection through the lymphatics, and it also transports dietary lipids. This is seen in your intestine. Then you have types of lymphatic tissue. You can have diffuse and nodular, so we'll come into a picture of this, or you can, and then, yeah, we'll come into the picture right now, actually. Ooh. Sorry, I don't think I have the picture in the next few slides, so I'll come back to it. But essentially, diffuse tissue is like it's spread out. So say this is your tissue, and the lymphatic tissue, it's just like spread out. There's like dots. It's completely packed, but it's all over. As compared to lymphatic tissue that's nodular, you would have them in areas. So this can be areas of lymphatic tissue in certain places. So you can see this in the intestine, for example. Then you have lymphatic organs. Lymphatic organs can be primary or secondary. Primary is where immune cells develop. So for that, you have the bone marrow and you have the thymus. Bone marrow for B cells, thymus for T cells. Secondary is where secondary organs is where like immune cells can come to fight off infections or antigens and so on. So that's lymph nodes, your spleen, and your tonsils. Very important to know these organs. Then you have a supporting network in your lymphatics. So this supporting network is present in your lymph nodes, it's present in your spleen, lymphoid tissue, but it's not in the thymus. Important distinction, it's an exception, so exceptions are always good to know. This supporting network is made of reticular cells, so type 3 collagen. And yeah, this is a picture of it. Remember, reticular cells, they have this appearance. Reticular connective tissue has this appearance because of its silver stain. And here you can see the reticular cells in this picture as well. Then we come into lymphatic vessels. So these are the ones that are doing the majority of the work. So these are thin-walled capillaries. And then this is where the difference is very important between a lymphatic capillary versus a blood capillary. So these features, very, very important to note, very easily a question. It could even be an SEQ named that lists the differences between lymphatic vessels and blood capillaries. So no tight junctions. So blood capillaries can contain tight junctions, but lymphatic vessels don't. They're very leaky. And then you can have fine filaments, which anchor the vessel to the surrounding tissue. Yeah, so blood capillaries don't have that, and lymphatic vessels essentially have filaments that are attaching them to the surrounding connective tissue. And lastly, their walls are more permeable, and this goes in hand with the no tight junctions. So you can see lymphatic vessels below the epithelium of the skin, and there are certain locations where they're not present, which is the cartilage, the bone, and the CNS tissues, so central nervous system. Moving on to the lymphatic vessels, this is a better image to observe them. You can see that this is your arterial, and it has a pretty solid like lining. So does the venule. But the lymphatics is like it's disturbed. Like here you have squamous cells all over, but this is mostly disturbed. It's very like leaky. There's no tight junctions between them. What they do is again they remove the substances and fluids from the extracellular spaces. So um, whenever fluid like leaks out and into the extracellular matrix, they take it in to prevent edema. And now with the fluid, when it's inside the vessel, it's called lymph. And so the way lymph flows, and we'll come to this even later, is they go from the lymphatic vessels to the lymph nodes. There in the lymph nodes, which are the secondary lymphatic organ, the antigens are filtered out, and then they are returned to blood vessels eventually. Okay, so the way this works is this is an image of your lymph node. Here you can notice the hilum, which is the point where all the like vasculature is entering from. So artery and vein is entering here, lymphatic vessel is entering here, and they're also entering here and here and here. So lymphocytes, which are immune cells, they can be in the blood vessels and they can also be in the lymph. So they can enter a lymph node through both passages. So you can have the lymphocytes entering the lymph node from the efferent lymphatic vessel, or you can have them entering through the blood, through specifically high endothelial venules or post-capillary venules. We'll come into this as well. So yeah, they can enter through HEVs or efferent lymphatic vessels. This is how lymphocytes can enter the lymph node. And then the entire process of filtration happens, and then lymphocytes exit the node through the efferent lymphatic vessel, the flow out of here. And then they can either go to the right lymphatic trunk or the thoracic duct, which are important lymph node lymphatic key points. They can go to either of these, and eventually they will still end up draining into the normal blood circulation because you're taking the extra fluid that was in the extracellular matrix and you're returning it back to the blood because that's where it leaked out from in the first place. So back to the high endothelial venules. This is what they look like. Okay, these are what you need to know. They're cuboidal or columnar in appearance. Um, you can kind of tell, but not really, so just memorize this point. And they're found in the paracortex of the lymph node. And we'll get into this as well. Oh, this is where the picture went. Yeah, I think it just got disorganized in the slides, but this is what diffuse versus nodules of lymphatic tissue look like. So diffuse, this entire thing is lymphatic tissue. It's spread all over. Versus nodules, you can have like you can have little patches of lymphatic tissue coming together, and they can be in bunches, and these can be called Peyer's patches, for example, which are found in the intestine. Yeah. Then we come into the structure of the lymph node. So, as we looked before as well, the lymph node looks like a bean or a small kidney, and it has the hilum for the neurovasculature, so and the lymphatic vessels as well. So if you're in lymphatic vessel enters through the hilum, vein and artery enter through the hilum, and all around it you have the efferent lymphatic vessels as well. So lymph nodes essentially, because they come along the pathway of a vessel and they're spread out all over the body, and they have access to blood flow and they can go to literally any organ, they're a risk for metastasis. So if cancer spreads and it's going, if a cancer gets to your lymph nodes, it's very high risk for it to metastasize to literally everywhere because it's traveling in the lymphatics. Yeah, and so you have capsule here, and an important point to know for lymph nodes is that the flow that's coming into the lymph nodes, it's unidirectional. It's not going to come down the same path and go up the same path, and that's because it has valves. That's similar to how we have semilunar valves in the veins. And again, the purpose of lymph nodes is they're the secondary lymphatic organs, so that's where lymph comes for filtration, for microbes, and so on and so forth. And they're also where immune reactions can take place. So lymph nodes, type 3 collagen, again, very important to mention. So we mentioned type 3 collagen is for vesicular fibers, and it's also involved here in lymph nodes. So lymph nodes can have a cortex area. So this entire thing is cortex. They have a paracortex, which is below the cortex, and then they have the medulla. So the cortex contains the lymphatic nodules. So here's a good place for it. You can see like it's a collection. It's not spread out all over. It's a collection of lymphatic tissue. And these can be primary and secondary nodules. Then you can have the paracortex, which does not have any nodules. So you don't see something like this here. It's pretty clear for most of it. And it also has the T lymphocytes. And then lastly, you have the medulla over here. So this contains the medullary sinuses. So these whitish spaces that you see, the lighter ones, that's the medullary sinuses. And they also have B lymphocytes and macrophages and dendritic cells. So the immune cells are in the medulla. Here we can come closer in, and here you can again see these are the sinuses, the open spaces through which blood travels. So when we come to the sinuses, there's three main types. There's subscapular, trabecular, and medullary. So what's happening for the sinuses is we have the lymph fluid coming in. Sorry, we have the lymph coming in to the from the efferent into the lymph node, and now they have to pass through the to the efferent, right? So the passage of the lymph from the efferent to the efferent, this is guided by the sinuses. So you start from the top and you go all the way down to the medulla, which is, and then the bottom of it is where you have the hilum. So you have subscapular, which is right below, not subscapular, subcapsular. These are below the capsule. So these are the ones that receive the efferent vessel. Then you have the trabecular. So you come down and you can notice in this area, this over here is the trabecular sinus. It's marked. It's a continuation of the subcapsular, but it's going down. And it's right below the cortex near the paracortex area. Then you have the medullary sinuses, which are just a continuation of the trabecular sinuses. So again, subcapsular, trabecular, medullary, the three sinuses that we have. Around the sinuses, you have a large variety of macrophages and immune cells. And so what happens is, as the lymph is flowing through the sinuses, their movement is slowed down. So the lymph is slowing down, and some macrophages can attack whatever microbe is there at that point. And then this again is another image to show you the sinuses. Then we come to lymphatic nodules. So remember we mentioned there's primary and secondary nodules. So there's a difference between them. So okay. So the way this works is primary nodules, they don't have a germinal center, and secondary nodules do have a germinal center. So let me see. Here, this is the primary nodule or the primary follicle, and you can see that it's very like one colored. It only has one color to it. Let me see if I can remove. Okay, wait. Mm, Okay. So notice again, the primary follicle, it only has one color around it. So it's very like dense in one area. It doesn't have a germinal center. So it doesn't have B lymphocytes which are maturing in any such thing. They're only inactive. And then you have the secondary follicles, which you can see there's a dark outline, and then there's a center which is lighter. So secondary follicles. Or the germinal center, which is also visible here. So it's a lighter area essentially as compared to the outer part. These contain the B lymphocytes, and that's where they mature and they can differentiate. And so the area that surrounds them, so this is your small follicle, this is the germinal center, this area outside it, this is the marginal zone. So for germinal centers to develop inside your lymphatic nodules, you need to first be born. They can occur when you're still like in utero. They won't occur if you're still an embryo. They will occur after you're born, and they need a stimulus. So an antigen should be exposed to the body, and then you would develop germinal centers, which is, you can think about it. B lymphocytes aren't going to proliferate and differentiate if they don't have something to work against. And again, this is a picture, sorry, of B lymphocytes versus T lymphocytes. There's not much explanation given behind these pictures. I would just say memorize it. You can see for B lymphocytes, it's like one solid color in the nodule versus for T lymphocytes, it's outer dark and inner lighter. Yeah. And then we can move on. Then we come to the thymus, which is a primary lymphatic organ for the maturing of the T lymphocytes. And the T lymphocytes, this is important for adaptive immunity. Now, the cells that you can find in the thymus, they are thymocytes, epithelial reticular cells. So thymocytes, you can see here, they're like all the dark cells. That's the packing of the thymocytes. Epithelial reticular cells, they're these ones. So they're like lighter cells that you can find in between the thymocytes. We also have B lymphocytes in thymus, they can come there. You can have macrophages, mast cells, and APCs. So APCs are antigen-presenting cells. And for the supporting network, here, as I mentioned, lymphatics, their supporting network is made by reticular cells, type 3 collagen. So they are joined by desmosomes and tight junctions. And they form the capsule of the thymus, and then the capsule goes down to form the little septum. There's also something called cytoreticulum, which is where macrophages and lymphocytes attach. And this is pure memorization. So we come to the capsule of the thymus. Here you can see the capsule. It's going down and it's forming the little septae, and these are also known as trabeculae. And then you have the cortex of the thymus. So this entirely is this entire thing is the cortex over here, and you can see it here as well. The cortex contains the thymocytes, so these cells that we mentioned, the cortex is just packed with them. It looks very dark, and these are the immature T cells. They're waiting here for maturation, and they contain they don't contain the reticular fibers. Then you have the medulla, so below the cortex. Here you have something called Hassel's corpuscles. Um, they look like this. They're like little pink, circular, blobs. And you can see that they're circling inside. So these are Hassel's corpuscles. Their function isn't known, but it's like theorized to be linked to interleukins 4 and 7. Also, just keep ones like to memorize together in case they do come. They don't contain any afferent vessel. They only contain efferent. This can again be a recall question. Not too much critical thinking. Again, they can just say which one of these is true about a thymus, and this could be your answer. Then we come to something called the blood-thymus barrier, and this is something that is the blood-thymus barrier. Essentially, when you have T cells that are developing inside your thymus, and you don't want to expose them immediately to antigens when they're still in the developing stage. So this protects them from any antigens that are traveling in the blood. It's also protecting the blood from any like rogue T lymphocytes which haven't been developed correctly. So it's a barrier. And so it's made, what this barrier is made of is these 4 components. Very important to note, in my opinion. Capillary endothelium, so they have tight junctions. So it's a barrier, it's a full barrier. A thick basal lamina, and then they have perivascular connective tissue, and they have macrophages in case antigens do escape into the thymus. And they also contain type 1 epithelial reticular cells, which is your final protective layer. So they can assure that these can assure that the T cells get to mature and grow properly without being disturbed by antigens. And so this is important just for the T cells development, so they later on don't cause problems. Then we come to another secondary organ, tonsils, and tonsils are located in a specific pattern. I'll get into it. But essentially, tonsils have lymphoid tissue, and they can protect you from any pathogens that you might like eat, because they're in that essential area. You can also have, so there's 3 types of them. You have palatine, pharyngeal, and lingual. These are the 3 types of tonsils that are mentioned in your lecture, and they are said to be arranged in Waldeyer's ring. So what that means is they kind of look like this. This is like when you open your mouth, it kind of gives this appearance. You would have your pharyngeal tonsil right around here. You would have your lingual tonsil here, and you would have your palatine tonsils here. And then here you would have another group of tonsils. So this is pharyngeal. You don't need to know this arrangement. This is just for your understanding. 1 second. And here, lingual. This is your Waldeyer's ring, sorry. So again, these tonsils are arranged in this form, and they're protecting you from any pathogen that you're ingesting or taking in, or any pathogen that can enter into your body while breathing, because same pathway. So this is what palatine tonsils look like. You can notice that they are non-keratinized epithelium, so they're related to non-keratinized epithelium, which is, yeah. Then you have, they also contain tonsillar crypts. So again, just keywords to remember. Then you have the pharyngeal tonsils. So essentially they come right under, okay, so um, this is your marking for pseudostratified epithelium, and the pharyngeal tonsils come under it. And so that means like their lymphatic tissue, or yeah, their lymphoid tissue comes under the pseudostratified epithelium. And lastly, you have the lingual epithelium. So this is the lingual tonsils. These are under your tongue. So your tongue's epithelium is stratified squamous. This is the epithelium. Under it, you have the lymphoid tissue. You can see them very clearly. And these also contain crypts, just like the palatine tonsils do. Oh, sorry, and these are the circles are the lymphatic tissue, and the crypts are these lines. Um, you know what? Just for the sake of understanding, I'm gonna go over this one more time, just in case. Palatine tonsils, again, all these tonsils, they contain lymphoid tissue for any airborne ingestion or airborne or ingested microbe. You have crypts here, which is just a feature to know, and these are your lymphoid tissues right here in the palatine tonsils. They are related to non-keratinized epithelium because of their positioning. Then you have pharyngeal, so this is your marking for the pseudostratified epithelium. Pharyngeal tonsils are related to this, and under it is the lymphoid tissue. Then you have your lingual. Lingual is under the tongue stratified squamous epithelium. Right here, under it, you can see the lymphoid tissue in the lingual tonsils, and they also have crypts. I hope that is clear. An important point to note is they also lack afferent lymphatic vessels, just like the thymus. Then we come to the last organ, hopefully, which is the spleen, and then we'll just do a quick overview of the immune system. So last organ we come to the spleen. It's the largest lymphoid organ that we have, still secondary. It filters blood and it's filtering blood, it's removing old RBCs, and it's also a site for immune reactions to take place. So the structure of the spleen, it has a dense connective tissue capsule, and this capsule contains myofibroblasts, which I mentioned are similar to muscle cells, but also connective tissue cells, so they can contract. So they contract and they can help release blood cells into circulation when needed, because you are filtering blood, you're removing old red blood cells, so you want to release the replacement. Then this also contains a hilum, which is the entry point and exit point for the lymphatic vessels, as well as the nerves, arteries, veins. Then lastly, you have the stroma, which is a meshwork of reticular fibers. Again, type 3 collagen, very important, as I mentioned. Yeah. Okay. This is what the capsule looks like. You can see it's dense connective tissue. It's very, very packed, and it's very clear. Here you can kind of see it here if you zoom in, but the other two pictures are clearer enough for you. This is what the stroma looks like. Again, it's similar to how the reticular connective tissue picture looked like. It's because of the special stain, you can pinpoint it very easily. Then we come into a specific important part of the spleen, which is the splenic pulp. So this is essentially the functional tissue of the spleen, the part that's doing all the functions that we did mention. So we have the white pulp and we have the red pulp. So white pulp, okay, let's come here. So in this picture, everything is mostly pink, and then you see the little blue dots here. This is the white pulp. This is probably because of the dye that it's colored blue, but the white pulp essentially, it contains, the white pulp is what's doing all the immune reactions, okay? Because we mentioned that the spleen is still carrying out immune reactions, and the red pulp is the one that's associated with the blood filtering and everything as such, and it kind of works, right? White, related to white blood cells, think of those, think of immune reactions. Red, blood is red, so it's related to the blood part. So, white pulp contains peri-arteriolar lymphoid sheaths, or PALs. So these are T cells that are surrounding central arterioles. I think I should have a picture of this. If not, then I would recommend searching it up. The lymphoid nodules, these are, they have lymphoid nodules in the white pulp, and this is again where you have the B cells. And then function, as you mentioned, immune responses. Then we have the red pulp. So the red pulp contains sinusoids. Sinusoids is essentially places where blood can flow through. And so that's what the sinusoids are. So because they're carrying out the blood part of the speen's function. And they also contain splenic cords, something called cords of Billiroth is important here. They contain RBCs, macrophages, and lymphocytes in the red pulp. The red pulp also has something called stave or stave cells, and they have a function, and we'll get into them. So this function of the red pulp is, again, filtering blood, removing old blood cells, and so on. And they can also have macrophages in the red pulp just to fight off any infection that's coming in from that side. So this is a picture of the, okay, look here first. You can see majority of this is red, and then you see this area. This is the white pulp. Here is the central arterial that we mentioned, and this is the white pulp surrounding it. I'm sorry, this is the pal surrounding it. And then we come here, these are the sinusoids, the clear spaces through which blood can run through in the red pulp. Then we come into splenic circulation, so like the blood flow of the spleen, the blood flow of the spleen. So essentially, you start off with the splenic artery, branches into trabecular artery. Think of it like capsule. Capsule came down to trabeculae. So splenic artery coming down to trabecular arteries. They enter the white pulp. They make those central arterial. Central arterial is surrounded by pal, and then from the central arterial, you get smaller vessels. And now these smaller vessels are called pennicellar arterioles. So just yeah, pennicellar. Just think small. I don't know. And now these small pennicellar arterioles are the ones that are going into the red pulp. So white pulp has central arterioles, red pulp has pennicellar. So yeah, that is the essential flow you have of the arteries. Then we have some circulation pathways. So splenic circulation has two parts. It has a closed circulation and it has an open circulation. So closed circulation essentially means they're going from capillary into sinusoid and they're within vessels. Like this is the main part. They're enclosed within vessels. They're not moving into any open space. Versus open circulation is blood from the capillaries. It comes into the splenic cords. So remember the splenic cords that we showed here. And then they have to enter the vascular system again. So the blood is now out of the vessel. It's in the splenic cords, and it has to, as it passes through the splenic cords, it has to later enter back into the vessels. And it does this by squeezing between stave cells, and it goes back into the sinusoids. I hope that made sense. So what this does is, as you mentioned, the spleen's function is to remove old RBCs and so on. So in this process, when blood is now in the open space and it has to enter the vascular system again, and it has to do so by squeezing, only the healthy cells, the normal cells, which are still of good health, will be able to carry out this procedure to go back into the vessels, while the damaged ones are going to be removed by the macrophages. Important point to note that when the RBCs are digested and their iron is released, this is stored by the macrophages. Lastly, you have the venous drainage. So we talked about arterial, we talked about the pathways inside, and then we're going to talk about venous. They drain into the trabecular vein, matches the trabecular artery, and exit the spleen through the splenic vein, matches with splenic artery. That is splenic circulation done. I think this should be the last slide. This is linked to immune system. So how, like just like a summary of the immune system, you definitely know more of this through HLS. So you have innate immunity and you have adaptive immunity. Innate immunity is the one it's not specific, it's really fast, it's at the very start, and it usually involves the cells like macrophages, neutrophils, natural killer cells, and innate immunity plays a bigger role in inflammation. Then you have adaptive immunity. So this is where, this takes time to develop. This can regenerate again later on. And this is a special response directly for the microbe or the antigen that's being targeted. And this involves the cells like B and T lymphocytes. Adaptive immunity has two further parts. It can have humoral or cell-mediated. Humoral is for B cells, cell-mediated for T cells. Yeah, that's immunity covered. Then an important cell to note is the dendritic cells, or the antigen-presenting cells that we mentioned before as well. These are the most important APCs, some examples of them, some names that you should know. Langerhans cells and follicular dendritic cells. These are in the germinal centers in the lymphoid follicles. Yeah, and with that, we are done. We are done with all lectures. And now we're coming to just some questions for this topic. And yeah. So the following is a primary lymphoid organ, which cells are primarily found in the germinal centers, thymus, and B lymphocytes. Moving on, which type of lymphatic vessel enters the lymph node to bring lymph for filtration? Yeah. And then what is the primary function of high endothelial venules in lymph nodes? You can pause. I'm going to move on. Afferent vessel, always know the difference between A and E. Don't miss this exam question. Don't lose this mark. And again, high endothelial venules, therefore entry of the lymphocytes into the lymph nodes. They're not draining it. They're not removing anything. They're just bringing the lymphocytes to the lymph node. Yeah, and with that, we are done with all three parts: epithelium, connective tissue, lymphatics. I hope this PAL session was beneficial to you. I wish you all the very, very best on your exam. Inshallah, you'll all succeed. If you have any questions, feel free to contact me. I'll do my very best to help you. And yeah, and that is it. I wish you all the best. Assalamu alaikum.